Aaron,
Hi, Ilya. What's up?
so you're in China now?
I'm at Stanford.
Oh, Stanford. Okay, that's cool.
I was in China when we were in that meeting together.
So traveling is okay, back and forth.
It's not easy, but it's possible.
It's possible. Yeah. I'm going to Korea in January, so I want to know how that. Korea, I think it's easier.
Yeah, Korea's fine.
Aaron.
Hey, Alejandro. Hey, Alex. Good to see you.
I see you too, dude.
How's it going?
Chugging along, still getting the lab oriented.
I was chatting with Neil Schneider about you.
Yeah, I love Neil. He's a great guy.
Yeah. Yeah, he's gonna win the Nobel Prize, I think.
For the ASO stuff?
Yeah.
No, it's really cool. It's really impressive what he's managed to do.
I don't know if you've seen the latest, but it looks like it's working.
Oh, no, I only saw the bits of the nature medicine stuff. I haven't seen any super later than that.
There's a kid like in Germany who he got to very early on, and like it's just like completely stabilized. These are all FUS mutations, which are very aggressive.
That's amazing.
Yeah.
It also is a good proof of concept for the whole field, right? Like one thing people keep saying about neurodegeneration, you got to get in earlier and earlier and earlier. And if you actually now finally have the proof in the pudding, that is key. That's pretty good to see.
The SOD1 stuff is also supporting that. Like that's working now. That they're going longer and longer also.
Cool.
Do you want to test out your slides?
Yeah, let's do that.
I was actually just back at Penn last week.
Nice. I miss Penn, man. That was a good place. Do I need to switch it?
Yeah, looks good. Yeah, looks good.
Great.
Yeah.
Cool.
Hi, Dr. Gittler.
Hi, Husniye. Good to see you.
Good to see you too. How are you?
Great. Thanks for joining.
Thanks for inviting me. Should I test if my presentation is working?
Please.
How does this look?
We just see your like web browser. So I think you have to share or go to your either just share your PowerPoint or go to it.
Is this not working?
Yeah, it looks good. Perfect. Yeah.
Okay. Thank you. Cool. Thanks.
I bet you're liking New York. I mean, San Diego. Alex.
It's lovely. It's lovely. Yeah, it's just a little different from New York, needless to say. I'm not like walking 40 minutes each way to and from work, and it's glorious. I have no problem doing that.
I was just hanging out with some Columbia colleagues in New York.
Who in particular? Just I guess Neil sounds like is one of them.
Yeah, Neil was at the meeting, but Yamamoto.
Oh, I love Ai. She's awesome.
And then some guys from Cornell.
Cool.
Asa Belyevich. He's sort of got kicked out of Columbia, but
How could I not know Asa? I took over his lab space. I thank Asa for inheriting many pieces of equipment.
Yeah. That guy did amazingly well because, you know, despite getting kicked out, he sold his company for, anyway, he has a nice apartment because of that.
He's good for him. Good for him. Did you know Asa I guess from a long time ago, Aaron? Or
Yeah, we go way back.
okay.
Yeah, there was a neurodegenerative disease Cold Spring Harbor meeting. Yeah, you would have fit right in there. A lot of CRISPR screening stuff and this and that.
Yeah, it's nice to see how technology like CRISPR is really starting to move its way into this field. There's a lot of really interesting screens people are doing.
I'll be out there in San Diego January-ish.
It's on my calendar, man. I'll be there. You just let me know. Yeah, I'd love to hang out.
I'm also gonna secretly come down there at the beginning of January. Honestly.
Well, if you want me to let me know when you're secretly around, that's cool too. Yeah, no, it's love to just kind of catch up again. Be really fun.
Yeah, sure.
Are you going to keep organizing the Neuro Zoom? It's like the long-term vision here.
I think so. It kind of keeps going.
I think it's great. It's a great way to get people from around the world to hear each other's science. I think there's not enough of that at that scale that you've managed to do it at, which is really great.
Yeah, that was like a good byproduct. I was just me bored wanting to hear some science every once in a while. Yeah, we have these like cool Neuro Zoom swags.
Oh, nice, nice. How does one attain the swag? You need to organize 50 meetings?
Like when you know, like.
Sounds good, Aaron.
Is Brad Johnson still going?
Yeah. Brad's doing great. Yeah, he's still at Penn, doing a little bit more clinical stuff, but the lab's going well too. He's got some interesting stuff going on. Still looking at like some aspects of telomere biology and senescence, and also Wnt signaling.
Nice. Right in the middle of between like Aubin Pond, BRB and Abramson, where Aubin Pond is, they're building a huge hospital in that courtyard, a CHOP building.
Really?
That's all blocked off now.
Yeah. Penn's just gone out of control with their construction. God bless them. It's amazing.
And then they got this $100 million patent royalties from the Pfizer vaccine. So they have this RNA Institute.
Oh, wow. Good for them. Yeah, it's good for Penn. It's a good place. It's a good place.
Yeah, I feel like when I come back, even though I've been out of Penn for over 10 years. I feel like when I go to Aubin Pond, they still remember me. Like people in the coffee shop here at
What?
Stanford don't even know me.
Wow. Yeah, I think people at Penn really all knew each other. And that was really nice. It's hard to take that for granted. Yeah, I think I took it for granted. Now I kind of realize it's not super common.
I went back to my PhD lab and like they were all running over like, oh, we found a box with your name on it and stuff like that. Are you meeting people from UCSD?
Yeah. Yeah.
Okay, that's
I already have a couple of really nice collaborations going on. Some of the stuff with Susan Ackerman. She's been amazing. Really good collaborator.
Good, yeah.
Bing Ren's been amazing. Jean Yu. It's a really good group of people here.
Nice. Yeah, there's some good UCSD NeuroZoom fans. We're still waiting for Zilong.
Okay.
Just a minute or so.
Oh, I'm here. Sorry. Aaron, sorry.
Hey, Zilong.
Park my car.
How are you doing?
Alex.
Hey, what's up?
How are you?
Good.
Nice to see you. Yeah, I'm getting to the office in a couple of minutes.
Nice. It's the great thing about Zoom. You can do it on the move.
All right, I'll try to delay a little bit. So
It's all right. It's all right. I'm getting the elevator in a couple of minutes.
okay. You're in Shuhui?
Yeah.
Okay.
You might heard the news. I'm leaving Ireland.
I was just in China last week, so it's good to be back in California.
Sure. Yeah, I'm going to get started.
Okay. All right. Oh, you mean? Hey, you mean? We're twins today.
Yes. Yes. Thank you for the,
Yeah.
very warm, very nice. We need it. It's pretty cold in San Diego these days.
Yeah. Alex was asking how to get the hoodie. I was just saying, like, you don't really ask for them.
That's right. That's right. Yeah.
Okay. On that note, let's get started. Hi, everyone. Welcome to a Stanford edition of NeuroZoom back in the Bay Area. Looking forward to hearing from Husniye and Alex on their exciting talks. But first, let me advertise next week's NeuroZooms. We have talks from UCSD and UCSF, two outstanding public universities in California. Looking forward to hearing the latest from Xin Duan and Gulchin, and please tune in. Let me or Zilong know if you want to present your work. You can go to the YouTube channel, you can subscribe, looking for speakers, postdocs, students, PIs, everyone's welcome. And with that, Zilong, are you able to introduce Husniye?
Just a second. Getting there. I thought I introduced Alex.
I'll introduce him. Okay. I can introduce Husniye.
Oh,
sorry.
Yeah, thanks. Sorry.
Yeah, no problem. So it's a really great pleasure to introduce our first speaker, Husniye Kantarci. She's a superstar postdoc at Stanford, working with in Brad Zutro's lab, and she's hit the ground running and made a really exciting discovery that she's going to tell us about a really unexpected role for Schwann cells during sensory neuron development. She's just an amazing scientist, and you're going to love this story. Husniye, take it away.
Thank you so much, Dr. Gittler, for this super kind introduction. Share them. Can you see my screen?
Looks great.
Okay. Oops. Sorry. Thank you so much for this opportunity to give this talk here. As you said, I'm really excited to tell you about our discovery of a novel role for Schwann cells in the nervous system in promoting excitability during development of sensory neurons. I'm a developmental biologist by training, so when I started my postdoc, I have been really interested to understand how the cells develop into excitable neurons that are capable of firing action potentials. It's usually thought that the neuronal differentiation program autonomously allows for development of this function. But we asked the question, are glial signals actually required for development of excitability in the nervous system? Glial cells are non-neural cells, as you mostly probably know, that constitute more than 50% of the cells in the nervous system, and they actively regulate many aspects of neural development, including myelination and synaptogenesis. But whether they play a role in promoting excitability, which is the most basic function of the nervous system, is largely unknown. We wanted to answer this question using dorsal root ganglion neurons, which are the sensory neurons. Their cell bodies are located next to the spinal cord, embedded in the spinal column, and they send long projections to the periphery of the body, and they give information related to our sensory environment. The DRG neurons have multiple subtypes that are capable of relaying a variety of sensory modalities, including pain, touch, itch, temperature, and proprioception. All of the DRG subtypes are in close interaction with Schwann cells. Their long axons are either ensheathed or wrapped by Schwann cells in the periphery, which forms this great system for us to study this glial neuron interaction. So if we look at closely to the DRG neuron development, in mice in embryonic day 11, that's when they first coalesce into the ganglia. And at this stage, they are postmitotic, but they're not excitable. By embryonic day 16, they start showing some spontaneous activity, which at later postnatal stages is completely replaced by high sensory evoked responses. That's enabled by an increase in expression of the voltage-gated sodium channels that endows neurons with action potential firing abilities. So Schwann cell precursors appear in the DRG ganglia at about embryonic day 12, and they start differentiating into myelinating or non-myelinating Schwann cells. So as I will also show you later, this puts Schwann cells to the right place at the right time to signal to the neurons for promotion of excitability during development. So to be able to answer this question, the glial signaling important for development of excitability, we wanted to separate DRG neurons away from Schwann cells during development and test whether they maturate correctly and capable of firing action potentials and become excitable. We achieved that by using an immunopenning method that my advisor, Dr. Brazuker, developed during his postdoctoral studies when he was a postdoc in Bambaris lab. By this immunopenning method, we were able to purify DRG neurons to more than 98% purity in cultures, and this really enabled us to test for the first time are neurons excitable in the complete absence of glial cells. So normally DRG neurons in vivo and in vitro in the presence of glial cells fire these beautiful action potentials. But when we separated them away from glial cells using this immunopenning method and culturing them in pure normal cultures, they were unable to fire these action potential trains. And even with the suprathreshold current injections, most of the time, they will only fire one action potential. We hypothesized that was because they're not interacting with Schwann cells anymore. So I separately cultured Schwann cells and collected their media, which is called Schwann cell condition media, which theoretically has all the Schwann cell secreted factors. And I treated DRG neurons that are alone in culture with Schwann cell condition media, and that restored action potential firing abilities of neurons. Overall, this showing that Schwann cell conditioning media actually promotes excitability and DRG firing. We repeated this experiment multiple times. Here, you're looking at each gray dot as one cell, and each colored dot is one biological replicate. And we consistently observed that treatment with Schwann cell secreted factors lowered firing threshold of DRG neurons, which suggests that they are more easily excitable. It also increased the number of action potentials fired by DRG neurons, overall showing that Schwann cell secrete factors that promote DRG excitability. What is the mechanism of this effect? As I said at the beginning, sodium channels endow neurons with action potential firing abilities, and DRG neurons express three major voltage-gated sodium channels, NAV 1.6, 1.7, and 1.8. When neurons cultured by themselves away from glia, they express very low levels of these sodium channels, which is consistent with the fact that they are hypoexcitable. When I treated them with Schwann cell conditioned media, we rapidly rescued this effect and saw that expression of the sodium channels increased by 2 to 4 folds in response to the Schwann cell secreted factors. We saw that a similar effect was observed at the protein level as well. So not just the mRNA, but also protein levels of these sodium channels increase with treatment with Schwann cell secreted factors. So overall, this brings me to my first conclusion that Schwann cell secreted factors that promote DRG excitability by increasing expression of voltage-gated sodium channels. What is the factor that is coming from Schwann cells that has this important function? Through fractionation and mass spectrometry, we identified that Schwann cell conditioned media was very enriched in prostaglandin E2, which is a small lipid molecule that has already known to have prominent roles in inflammation and pain. So we hypothesized that the excitability-inducing molecule in the Schwann cell conditioned media is PGE2. And I did a battery of tests to actually test whether PGE2 first is sufficient to have this effect. So first, I treated cultured DRG neurons with prostaglandin E2, PGE2, and similar to Schwann cell conditioned media, PGE2 as well increased expression of sodium channels NAV 1.6, 1.7, and 1.8. Also similar to Schwann cell conditioned media, PGE2 treatment lowered the firing threshold of DRG neurons, showing that neurons are more excitable. It also increased the number of action potentials fired by DRG neurons, overall showing that PGE2 is sufficient for this effects of Schwann cells. Next, I tested whether PGE2 was necessary. I blocked PGE2 signaling in DRG neurons using two different methods. The first one is blocking PGE2 receptors, EP1, 2, 3, and 4, using chemical inhibitors. And we also use an antibody that neutralizes PGE2 molecules in the media. And both of the treatments completely remove the excitability-inducing effect of Schwann cell conditioned media, showing that PGE2 is required for this effect. And finally, I tested this mechanism in vivo. I injected PGE2 to the sciatic nerve where Schwann cells either wrap or ensheath DRG axons at a stage, at P0, at a stage where sodium channel expression begins, but it hasn't reached the postnatal adult levels yet. So in vivo as well, injection of PGE2 rapidly increased expression of sodium channels NAV 1.7 and 1.8, showing that PGE2 also has this effect in vivo. Overall, these results show that PGE2, Schwann cells secrete PGE2 to increase sodium channel expression in DRG neurons and promote excitability. We wanted to know if this was required in vivo, whether Schwann cells secreted PGE2 was important for neural development. So there are 4 enzymes that are capable of synthesizing the last step of PGE2 synthesis. And published databases show that Schwann cells express only one of these enzymes called PTGS3. I have confirmed that using RNA-scope, showing that PTGS3 is expressed by Schwann cells in the sciatic nerve. So we wanted to create an animal model to test whether Schwann cells secreted PGE2 was important for development of excitability in sensory neurons. So we generated flux alleles of PTGS3 gene, and I crossed this mice to Schwann cell-specific DHH Cre line to only knock out PTGS3 in Schwann cells and block their ability to make PGE2. So first, we tested whether these Schwann cells are impaired in their ability to increase DRG excitability. So just like my previous experiments, I cultured Schwann cells, this time from PTGS3 CKO mice. These Schwann cells are incapable of synthesizing PGE2. I collected their conditioned media and I treated DRG neuronal cultures with that conditioned media. So as I previously presented, untreated DRG neurons, when they're alone in culture, exhibit a high firing threshold, and they also fire very few action potentials with supra-threshold current injections. When we treat them with normal wild-type Schwann cell conditioned media, or the conditioned media collected from the control Schwann cells, the flux Schwann cells, we can rescue this effect and we can restore excitability to normal levels. But we don't see this excitability-inducing effects if I use the conditioned media from the CKO Schwann cells, showing that when Schwann cells are incapable of making PGE2, then they do not have this excitability-inducing effect. And you might be wondering, maybe this is because the CKO Schwann cells are somehow toxic to neurons. To test that, I added PGE2 into the CKO medium and show that that media does have the ability to promote excitability, lower the firing threshold, and increase the number of action potentials fired, showing that this effect is specifically due to the lack of PGE2 in the conditioned media from the CKO Schwann cells. Overall, these results show that PGE2 synthesis in Schwann cells is very important for the ability of Schwann cells to increase DRG excitability. But what is the effect of this in vivo? How does it affect in vivo development of neurons? First, I looked at expression of sodium channels in vivo. It's at the day of birth at P0. We observed that PTGS3 CKO mice, where Schwann cells are not making PGE2, express lower levels of sodium channels NAV 1.7 and 1.8. And this effect continues at least until the end of the postnatal stage. At P28, we still see a deficiency in the expression of sodium channels. And we do see a similar effect at the protein levels as well. At the protein level, NAV 1.7 and 1.8 are both decreased in the PTGS3 CKO mice, both at the day of birth and at postnatal stages. Overall, this data suggests that PGE2 secretion from Schwann cells is important for normal expression of sodium channels, but how does that affect neural activity? We in the lab do not have, at least I do not know the tools to actually measure neural activity in vivo. So we did something that's very close, or as close as we can get. I isolated DRG neurons from either flux mice or the CKO mice, and we acutely did this calcium imaging experiment to be able to understand whether neural activity was impaired in PTGS3 CKO mice. I induced neural activity by using this drug called veratridine, which is a sodium channel agonist. And we looked at neural activity by using a calcium indicator. So as you can see, these are the control neurons. Addition of veratridine, the sodium channel agonist, leads to a rapid calcium influx, suggesting that there's a rapid neural activity in the control neurons. However, we did not see this effect in the PTGS3 CKO neurons, showing that the sodium channel-induced neural activity is severely dampened in the PTGS3 CKO mice. So overall, Schwann cell-secreted PGE2 is required for initiation and maintenance of voltage-gated sodium channels in DRG neurons and normal neural activity. Which DRG subtypes are affected? As I said, DRG neurons have multiple subtypes that are capable of modulating different sensory modalities. So we wanted to know, is this affecting all DRG neurons or is it just some of them? To answer this question, because DRG subtypes are transcriptionally unique, we did a single-cell RNA-seq experiment from the neurons from the flux mice or the CKO mice. And we saw that two specific subclasses, CGRB neurons, which are the nociceptor that are capable of detecting pain and temperature, and proprioceptive neurons were specifically impaired in numbers. Since this could be due to the technical problems with the single-cell experiments, I did an RNA scope experiment to confirm these results. I labeled the CGRP neurons with calcitonin-associated peptides, or I labeled the proprioceptors with parvalbumin, and we did confirm that the numbers of these CGRP neurons or proprioceptive neurons were both increased by almost 50%. And expression of sodium channels in these subclasses was also impaired, suggesting that Schwann cell-secreted PGE2 is required for both survival of these neurons and normal expression of excitability regulating genes in these subclasses. So overall, Schwann cell-secreted PGE2 is required for the maturation and survival of these nociceptor and proprioceptive DRG neurons. How does this affect behavior in animals? So now we know which subclasses are impaired. I specifically focused on the sensory modalities modulated by these DRG subtypes. So looking at proprioception, with both rotor-rod tests, we detected a decreased fall latency, which suggests impaired coordination in this mice. I also did a skilled walking test and made the mice walk on a horizontal ladder. And we observed that PTGSC CKO mice were exhibiting more hind lip slips, which is again showing impaired proprioception. And with hot plate or Hargreaves test, I detected a decrease in the ability to tell the temperature in these animals, showing that Schwann cell-secreted PGE2 is important for sensation of temperature. This is also consistent with a loss of CGRP neurons in PTGSC CKO mice. And finally, I wanted to test whether these animals are feeling normal pain. So we did formalin injection assay. This assay gives 2-level responses. In its first 10 minutes, the response measures acute pain, which, as you can see, is normal in CKO mice, but the later developing inflammatory pain was impaired. So Schwann cell-secreted PGE2 is also required for normal development of inflammatory pain. With that, these results show that Schwann cells has this novel role in the nervous system in modulating normal expression of voltage-gated sodium channels and promoting normal neural excitability. And when Schwann cell-secreted PGE2 is impaired, DRG neurons in vivo do not express normal levels of sodium channels, do not have normal neuronal activity, and their sensory transmission is impaired. With that, some of the things that I'm really interested in studying in the future is testing whether glial PGE2 is actually important in formation of chronic pain, or is contributing to the neurodegenerative disease phenotypes. And I want to know whether Schwann cell-secreted PGE2 has other roles in DRG development, such as axonal development, or understanding how does it lead to these differences in DRG subtype development. And it will be really interesting to know in the future whether CNS glia also has this effect, and whether CNS glia also increase excitability during development. And just one teaser, a small experiment. I also collected conditioning media from oligodendrocytes and treated DRG neurons with oligodendrocyte conditioning media, and that also increased expression of sodium channels, suggesting that this ability of glia to increase excitability might be preserved in the central nervous system. If you'd like to read this more, our preprint is now online in bioarchives. With that, I want to thank Brad for his mentorship and this amazing project. I want to thank all of our collaborators, my lab, and funding resources. And thank you so much.
Awesome. Thanks so much, Husniye. Great talk. We're open for questions. Schwann?
Great talk. I have a question about where you think PGE2 is acting. Do you think it's acting in the ganglion itself or throughout, the Schwann cells in the periphery?
So our PGE2 injection experiment is to the sciatic nerve, and that gives like a rapid result of increase in expression of sodium channels. That suggests that PGE2 is acting in the periphery in the axons where Schwann cells are wrapping or sheeting DRG axons. There's also papers in the literature showing that EP receptors, which are the PGE2 receptors, are found in the DRG axons in the periphery, suggesting that, you know, PGE2 that is secreted by Schwann cells can be sensed by DRG axons. But I guess none of this eliminates that there's also something going on in the cell body. It might be both, but I think these experiments at least suggest that PGE2 in the periphery can be detected by the neurons.
Thank you.
So did you look upstream of the PGE2 synthesizing enzyme, like COX-2?
I haven't, but there's a lot of literature on COX-1 and COX-2 enzymes. So those are important for many different prostaglandins. So that's why we didn't manipulate those, because manipulating COX-2 might end up, you know, affecting other prostaglandins as well. So we focused on these last steps of PGE2 synthesis. But I think there's a lot of literature on how COX-1 and COX-2 are important for inflammatory pain and other things, but not on development of excitability.
Got it. Okay. David?
I might have missed this, but do you see any differences in morphology when you either with PGE2 alone or with the conditioned media?
In culture, overall, the cell bodies of neurons look very comparable. When you stain them with neurofilaments, they have normal, we haven't specifically looked at axonal development with proper tools, so I don't want to say there's like no phenotypes there, but the overall look of the neurons look very comparable. We don't see an overall change in morphology, but I think there's definitely reasons to like go deeper into that and do some experiments to specifically test whether axonal morphology is affected.
Cool, thank you.
David?
Hi, I enjoyed the talk. It seemed like in the knockout mice, some mice had very normal pain responses in the group. Did that correlate with the number of lost neurons in the DRG, or is it just complicated and there's other ways to compensate?
Well, that's a great question. We did see a different phenotype in males versus females. I can share that slide again. I do not know what is the exact. Okay, here, so the rounds are males and the triangles are females. So there is a female CKO mice feel less pain. We have talked about this to the pain experts and at Stanford, and they said there are already some literature showing like differences in inflammatory pain formation in males versus females, and there might be a reason for that we can look at like ASIC channels and other channels and how they are different between males and females. At this point, I do not know, but it is true that some animals are still developing maybe normal pain and some are like not developing anything at all.
But it's not related to the loss of neurons.
Could be. This pain response is modulated by CGRP neurons, and CGRP neurons are lost by about 50%. So it is possible that this is a response to the loss of neurons, but we are also showing that the remaining neurons are not expressing normal levels of sodium channels. So I think it is contributed by the both phenotypes. It's at this point hard to know which one is contributing to more, I think.
Thanks.
Okay, great. Thanks so much. It was great talk. Good luck with your paper. Zalong will introduce Alex, but first I'll give a intro to the intro, and then Zalong can introduce him. So Zalong will say about Alex's training and everything. But I just want to say, I first met Alex when I was just starting my lab as an assistant professor at UPenn. Alex was an MD PhD student, and we both worked in the middle of the night. And I mean, I worked in the day and night, as did he. And we just started to figure out how to teach each other how to do yeast genetic screens. And it was just a really transformative period in my career. And we figured out how to do it. And so all of the results from those yeast screens that I was doing in the middle of the night with Alex, all of those results are sort of what I built my lab on, and papers we're still publishing today. Now we've moved on to mouse and human and stuff like that, but all of it started just figuring out how to do yeast screens with Alex. So it's super exciting to see him now as a famous MD, PhD, and a genomics expert, and he's going to tell us all this crazy stuff he's doing. But it's long. You can give the more typical introduction.
Sure. Yeah, as you can know, so how exciting Aaron introduced Alex Chavez to come to our Neuro. So I'm a great pleasure to introduce Professor, now it's Associate Professor in UCSC, Dr. Alex Chavez. So Alex is starting his career in I believe this bachelor degree in Northwestern, working biochemistry and genetics, and in PhD, as I mentioned. So Alex started an MD PhD program in UPenn, where Aaron was starting his legendary career starting from East. So after MD PhD, Alex is starting a very interesting, diverse career path. One is very residential in Mass General. Another is starting a postdoc fellow in George Church's lab, right in the booming age of CRISPR-Cas. So in the middle, besides of the residents and seeing patients, Alex also spent a lot of time working in the lab, so starting all the revolution of CRISPR. To name a few, Alex has participated in the development of new CRISPR tools in the transcriptional revelation, genomic stability, as many others. So after the fellow published lots of papers, Alex first started his career in Columbia. So also starting his lab as well as the seeing patients and pediatricians. After a few years at Columbia, Alex moved to East Coast and UCSC. So being same time in the pediatricians, so Alex at the same time also in his lab developing all kinds of fancy tools. Not only the CRISPR-Cas, also recently, you can see the title of this talk, Alex is ready to make some new screening tools, making high throughput screening for the neurodegeneration disorders. Okay, it's my great pleasure to introduce the Alex Chavez. Now, talk of Alex. This talk is about high throughput screening for the neurodegeneration disorder. Okay, welcome.
Thanks, both. Thanks both Zilong and Aaron. Let me share my screen. And I think this looks right. We're good, great. And so it's actually, it's great to have Aaron here, because the talk I'm going to give is actually kind of going full circle, Aaron. It's going back to our days at Penn. So it's not a CRISPR talk, actually. It's a yeast screening talk. And so, yeah, I'm Alex. And really a pleasure to be here today. And so a few conflicts of interest. I'm consultant for VOR and Prevail, and I'm also a member of SAB of AdGene, Fit Balance, and Algorithmics. Overview for my talk is I'll tell you about our work on multiplex disease modeling, and the goal is to identify broad-acting genetic modifiers of protein misfolding. So before I kind of get into our work, just want to get a little bit of background on cellular models. So I think we're all pretty aware that cellular models, including cell lines, iPS cells, and organoids, have really transformed how we do biology and how we study biological processes in the lab. One issue that we all face, though, is many of us study one line at a time, simply because it's kind of hard to grow multiple different lines and do all these different experiments in them. And that's okay, but there's some problems with that I'm sure we're all aware of. And that's every single patient's unique. And so what we're all doing, or a lot of us are doing, is we're studying one example, hoping it applies to all. So obviously we get the results and we test in other lines, but we're just like hoping that that's really going to be true. And in addition, we've all kind of had these things happen where we're growing our cell lines and suddenly these weird things happen. We get karyotype changes, other mutations, and suddenly this line is not behaving as we thought it was. And that actually can really influence our results and become really big headaches. And so kind of realizing all these problems, our lab decided a long time ago, it'd be great if we had some sort of new technology that let us study not just one model at a time, but maybe dozens at a time. And so we thought by doing this, we could control for things like model quirks, these karyotypic changes, these mutations, and also get a more comprehensive understanding of disease biology. And so as a first sort of proof of principle of multiplex disease modeling, we turned back to a very near and dear model in my heart, which is yeast models of neurodegenerative disease-associated proteins. And so in these models, you take a little yeast cell, you make it express a protein associated with neurodegeneration like TB43, alpha-synuclein, et cetera. And when you do that, the yeast basically don't like it, and they're very sad and they grow very poorly. You then can do a genetic screen looking for genes that, when manipulated, make the yeast cells grow better. And it turns out that a lot of this work's been done over the years, and many of the hits that people find in these yeast screens translate when you take the human ortholog and test them in a million contexts. Really makes this yeast a very valuable discovery platform. And so what's the problem with current approaches? While they've been amazing, there's lots of proteins one might want to study, but to date, not all of them have been screened because it's a lot of work. In addition, for many of these neurodegenerative associated proteins, there's familial variants. Yet how they affect disease biology is still not 100% clear. There's some idea that they maybe help the proteins misfold faster, but clearly in cases of things like OPTN, you can really get two different diseases, glaucoma or ALS, right? So these mutations are clearly doing something, and today still not totally sure. And that's important because ultimately we're going to do clinical trials with patients, and we want to know, are all TP43 patients the same or all FUS patients the same? And so actually doing screens at this scale on multiple different familial variants is going to be informative for that. So again, instead of doing one screen at a time, we want to do something that was parallel. And by doing that, identify shared pathways across models versus just being unique to some, plus get better biological insight overall into these proteins. So how is it that we're able to track multiple models at the same time? We use a very simple approach, which is DNA barcoding. So here are 4 little yeast cells, and here are 4 little barcodes. And what we do is we insert these DNA barcodes into the yeast cells, into the exact same location in every single cell, and we know the exact sequence we're inserting in every single cell. So going forward, when you see a different colored yeast cell, that represents a cell that we know the DNA barcode for, and we know its exact position in the genome. And so now what we can do is we can take these blue yeast cells, and we can make them express a toxic gene like A-beta associated with Alzheimer's. Take the teal yeast, make them express a toxic gene alpha-synuclein associated with Parkinson's. Black yeast get TP43 associated with ALS. Then as a control, we'll take these yellow yeast and we'll give them a non-toxic gene EYFP. If we then take that pool of cells and we induce the expression of all these different proteins, the cells that express toxic proteins deplete. The cells that express non-toxic proteins like YFP, they enrich. And we can basically take all those cells, explode them open, and just sequence out the little DNA barcodes. And by doing that, know what's happening and what their growth behaviors are. And so we can then go back to this initial population again, this time induce a genetic change along with inducing the expression of those toxic proteins. If we get something like this, it would suggest that whatever that genetic change was, it's able to rescue the growth of this TP43 model because it's now growing better than it was in the control condition. And so kind of cutting to the chase, we now pull about 35 what we call disease models. We test them against about 1,000 human proteins, and we've identified hundreds of genetic interactions from those studies. I'll show you a small little widget of this data. And so the x-axis are a subset of our models. On the y-axis are a subset of the different genetic modifiers we've tested. You'll see different colored squares. The darker the square, the stronger that rescue is. And this is kind of a quick snippet of the data. And when you look across all these different tests, we found one hit that was quite tantalizing to us. And that was this one right here, which is DNAJV6. And DNAJV6 is an HSP40 co-chaperone that works in conjunction with other HSP70s. And so why were we so interested in DNAJV6? We found it quite interesting because it actually rescued multiple models of ALS. So we had multiple RNA binding proteins implicating ALS that this chaperone seemed to manage to rescue. And so that's what our screen told us, but we needed to go back and validate that individually. And so we went back to the individual yeast model that expresses FUS. We measured its growth, and then we added M-cherry as a control protein that shouldn't do anything. And we set that level of growth to 1. If we then take the FUS model and give it DNAJV6, we see a marked improvement in growth. So that's good, it tells us our screen is accurate. Looking at both TP43 and HRMPA1, we saw similar rescue. So this is good. It shows that DNAJV6 seems to be a bona fide rescuer of what the toxicity caused by these RNA binding proteins. One question you might ask yourself is DNAJV6 is actually one of about 41 members of DNAJ family chaperones. And so maybe any old DNAJ family member could do this. So to get at that question, we went back to the FUS model, took a few other just random members of this DNAJ family, and then looked at which ones rescued. And what you can see is DNAJV6 is clearly kind of the superior rescuer from the family, and that held across the 2 other RNA binding proteins. That again, DNAJV6 is kind of unique. It's not just any old DNAJ protein does this. There's something unique about this protein. So that's all fine and dandy, but you're all probably thinking to yourselves, well, that's in yeast, and what does that have to do with human cells? And so to get at that question, we did a different assay. And so it turns out if you just overexpress these RNA binding proteins that are implicated in ALS in human cells, they'll actually misfold. And you can actually quantify the amount of misfolding by doing biochemical fractionations. And so what we did is said, okay, what happens when we overexpress DNAJV6 in the background of cells expressing these RNA binding proteins? And we quantify the amount of misfolded RNA binding protein. If we look at FUS and quantify the amount of it that's aggregated by biochemical assays, You see that if you express FUS and YFP, we'll set that as a level of 1, so that's our control level. If you express DNAJV6 instead of YFP, you see a very, really sharp decrease in the amount of misfolded FUS that we observe. And again, the same thing holds when you look at TDP43 or HRMPA1. You see that DNAJV6 overexpression drastically reduces the amount of misfolded RNA binding protein. So again, a caveat of these experiments is they're obviously overexpression. And so you might say, well, if you overexpress any chaperone high enough, maybe that causes rescue. So it's super physiologic, doesn't mean anything. So get at that question, we decided to go back into our cells and this time knock out DNAJV6. So let's remove the endogenous levels of this protein and ask, do the endogenous levels have an ability to modulate the folding of these RNA binding proteins? And so if you look at FUS and you quantify the amount of it that's aggregated, you take the control cells, set that at a level of 1. If you knock out DNAJV6 using CRISPR, you see a 2-fold increase. Look at TD43, HRMPA1, same sort of thing. You knock out DNAJV6, you see a marked increase in the amount of misfolded species. So this really kind of told us that even in human cells, DNAJV6 seems to be a broad-acting chaperone against multiple RNA binding proteins. So this then leads to the obvious question, how is DNAJV6 performing its function? And to get at these experiments, we had an amazing collaborator, Peter St. George Hyslop, who's just like the nicest guy ever. And he decided to kind of help us kind of get into this biochemistry. And so one thing that was known is that, I'm sure a lot of the groups are aware that many of these RNA binding proteins have this remarkable ability to phase separate. And this phase separation is thought to be part of this process that leads to their aggregation. And so if you look at a protein like FUS and you purify it, and then you put it into physiologic salt conditions just by itself, it'll form these beautiful little condensates. If you take DNAJV6 and you purify it and put it in physiologic salt conditions, it does the exact same thing. It also can form these beautiful little condensates. And if you mix the two together, they'll actually they'll commingle and kind of exist in the same local area. And so this is a little bit tantalizing. It tells us maybe DNAJV6 is kind of hanging out with where our targets are, but does that have an effect? Does this commingling mean anything? So to get at that, it turns out if you take FUS and purify it and leave it just kind of over time at the bench, it'll start to clump up and you'll form these kind of gnarly looking aggregates. But if you take FUS and you add DNAJV6 to it, over time, you don't see those clumps form. DNAJV6 seems to prevent that. So this really argues that there's perhaps a direct effect of DNAJV6 on these RNA binding proteins. So to dig a little bit even deeper into the mechanism, we decided to really go for it and do a deep mutational scan of this protein. So basically, we made all single amino acid changes within a critical region of the protein thought to be involved in its ability to form condensates and to interact with substrates. And we made all possible single amino acid mutants, which is about 2,000 mutants. And we tested them all against the FUS model. And so I'm not going to share all that data with you, but I'll share a couple of vignettes from that part of the story. And one thing is that it turns out I didn't tell you that there's actually patients out there that have mutations in DNAJV6. They don't have a neurodegenerative disorder. What they have is a very prominent muscular dystrophy. But if you look in their muscles, they have TDP43 aggregates, telling you that maybe there's some data there in humans that DNAJV6 actually is a physiological modulator of TDP43. And so it turns out our screening data, when we did this massive mutagenesis, it seemed to argue these patient mutations were actually loss of function or even neomorphic. And so we decided to verify those results. Again, going back to the original FUS yeast model, the express M cherry doesn't rescue. Add in DNAJV6, it rescues, cells are growing better. If you take DNAJV6 that has patient mutations inside of it, you see a prominent loss of rescue. And in fact, one of the patient mutations actually causes like a gain in toxicity. And if you look at a different RNA binding protein, TDP43, you see that in fact, all of the mutations seem to cause a slight gain in toxicity, arguing that perhaps these mutations are actually kind of neomorphic gain of function. In addition, from our huge screen of different mutants, we also identified something we thought was pretty interesting, that actually identified variants of DNAJV6 that actually showed enhanced activity. And so this is taking the FUS model, and this time I'm studying DNAJV6 overexpression in the FUS model at a level of 1. Turns out we found several mutants that seem to rescue even better than the wild type DNAJV6. And when we took those and tested them against another RNA protein, TDP43, they also rescued, arguing that DNAJV6 is pretty good, but it's not been perfectly engineered to be super efficient at this and it can perhaps be made better. So next steps for this project is we want to test DNAJV6 in an animal model of ALS, and we're doing that actually in collaboration with Neil Schneider's group. We also want to understand a little bit more about biochemically how it's performing its function. In particular, we're really interested in these enhanced variants. You know, what's making them enhanced? What are the properties that we're modulating to cause them to be better? We also want to continue to build better versions of this protein. And we did this huge screen. We have all these other hits and we haven't followed them up. And so we'd love to kind of go back and revisit them. In addition, we've only screened 1,000 human proteins. We have about a library of about 15,000. We'd love to, again, just take this to completion. And then finally, our lab's been working on some in vivo CRISPR screens using animal models of neurodegeneration. And we are thinking about maybe linking up these two projects. So summary for my talk. Described how you can use DNA barcoding to study lots of different models at the same time. Despite the scale that we're operating at, you still get hits that are real and reproducible. We found this interesting hit DNAJB6 that seems to be a broadly active chaperone, and that although it's quite potent, it can still be further enhanced using protein engineering methods. So I like to thank a few people. Obviously, I don't do work anymore. I'm just really grateful to be surrounded by incredible students. And this work was all done by one incredible MD PhD student, Sam Resnick. And without Peter, we wouldn't have been able to do any of the biochemistry. And so obviously we're super in debt to him. And obviously I'd like to thank all the members of my lab that make coming to work amazing every day in funding, since no grants, no research. That'll take any questions.
Hey, great. Thanks, Alex. Short and sweet. It's amazing how used for amazing valuable that each screen can still provide some amazing new functions. So David, thanks for this question. Please.
Hi, I enjoyed your talk. I worked on DNA-J homologs in yeast when I was a graduate student. So
I love it. Yeah, man.
It was called Sec63.
Yeah.
And my memory is that DNA-Js don't have intrinsic ATPase or necessarily chaperone activity, is that they generally work with the DNA-K homolog. And they act as exchange factors, if anything. Have you looked at exchange activity, or did DNA or HSP70 homologs come up in your screens that wouldn't make sense of this chaperonin like model?
Yeah, so what's interesting is DNAJs can bind to targets, and by doing that, they can actually modulate some of their properties, but they do often work in conjunction with HSP70, and they sort of like bring cargo to HSP70, help with HSP70 undergo its catalytic cycle. And so it turns out if you delete the DNAJ domain, which is required for interaction with HSP70s, we don't get rescue anymore. So we do think that in vivo, that activity is important for its actual rescue. But we do know, at least in vitro, its capacity to bind to its target seems to modulate its activity. We likely think it's a combination of both factors. In vivo, you've got to be able to bind to it and bring in the HSP70 to cause the activity. That's probably leading to our rescue.
But does your in vitro assay require ATP hydrolysis?
No, there's literally just DNAJ in there and FUS, that's it.
Yeah, that's confusing a little bit to me, I guess.
Well, DNAJs don't undergo the cycle. It's the HSP70 that has it.
Yeah, but who does the energy to refold, help them change who moves the bonds around, I guess?
Yeah, so I think in vitro, in the purified protein experiments we're doing, it's literally just like binding to it and preventing it from misfolding more. I don't know. I don't think it's refolding it per se, to be honest with you.
Or unfolding. Okay.
Kang. Hey, Kang.
Great talk. Thank you. Can you comment a little bit more on the effect of DNAJ6 on the phase separation of FUS? Because you showed that it's prevented clumping, but I wonder whether the clumping represents phase separation or aggregates.
Yeah, so it's probably preventing their aggregation. So we've done a lot fancier methods on it, like FLIM and nano-IR. And it turns out that what DNAJ6 seems to be doing, and this is like getting into the weeds, it seems to be actually locking these RNA binding proteins in a gel-like state. And we think that that gel-like state is kind of like an intermediate. It's preventing it from going all the way to being an aggregate. It's kind of locking, it's like transient gelatin. And so we do think it's actually preventing the aggregate formation.
Great, thank you.
Thanks. Aaron?
Hey, Alex, awesome talk. 2 quick questions. Have you tested if DNAJB6 works to sort of synergistically with some of the potentiated HSP104 variants Jim Shorter's developed that attack those proteins like FUS and TDP43?
Yeah, we have it. That's a really cool idea because we do think they're working in different ways. So it could be interesting to check that out.
And then I couldn't help but notice that on the row above the DNAJB6, you had DNAJB8 working against, or B9 working against TMM106B. Can you say anything more about that? That seems really interesting.
Yeah, so we made this library, I don't know, 2 or 3 years ago, and there is, TMM1, it wasn't nearly as hot as it is nowadays. And so we honestly haven't pursued it a ton, Aaron. It's interesting, like little feature that we kind of saw. But honestly, I can't really comment more than, yeah, I think it rescues it, but how, why, what's going on, not sure.
Yeah, I've assigned everyone in my lab to work on that protein now. So I think it's really hot. So I think it could be, your system could be really good for it. TMA 106.
Yeah.
Cool, great.
Okay, Alex, I have a quick question about the, do you think it's possible to, you mentioned the protein aggregation, the phase separation, it's also happened a lot in the synaptic densities, but I'm not sure how is a random library technology can be applied to screening maybe some stachytrophy, protein aggregation, dysfunction mutants. How do you think about that?
So sorry, so you're saying that there's some synaptic mutants that also aggregate? Is that what you're saying? And that
Yeah. So also phase separation happens a lot in the synapse, as thin gap and shrunk. Although I believe some protein disaggregation problems also happen in some psychiatric disorders or neurological disorders.
Yeah.
So also they still happen with alpha-synuclein, right? So but would this synaptic protein be conserved in the yeast? Or how would you address these questions if you use this with similar tech to it?
Yeah, so some of the other proteins we're studying aren't necessarily conserved in yeast. The idea is in general that whatever the mechanism of toxicity that's existing in human cells is translated into yeast cells. So you don't need to have a direct homolog for you to observe that conserved toxicity. So I think synaptic proteins or whatever protein you have, as long as it has a phenotype in yeast, ideally an easy one is one that influences growth. It's just very easy to see that. Then you can do a screen on it. You can actually do other phenotypes. You just need to figure out how you're going to couple your outputs in multiplex to the barcode.
Okay, to see whether the aggregation affects the normal growth of yeast.
Yeah, super easy to test. I mean, Aaron has done this 1,000 times, and we've done it a lot too.
Yeah.
It's really trivial. And then the other thing you want to know is that actually the growth is dependent upon your phenotype of interest, right? So you ideally want to make sure that you think that the toxicity is driven by that, not some other crazy neomorphic function in yeast cells.
Sure, great. All right. Any more questions? Okay, it's more questions. Great. Like thank two speakers today. That's a super exciting talk. Husniye and Alex. Great. Thanks so much.
Thanks, everyone.
Thanks. Husniye, thanks. See you all next week.
Thanks.
Have two more Neuro Zoom before a holiday. Thanks.
Yeah.
Bye.
Yeah.
Bye.
Bye, everyone.